Introduction to the conventional cytogenetic analysis.
Cytogenetics is a study of structure, function, and evolutions of the chromosomes. It was first described as Nageli, the Swiss botanist,
Asher called these thread-like structures in the nuclei of a plant cell as transitory cytoplasm,
or also known as chromosomes today. Later on, in 1888, von Welder described the terms chromosomes. Chromos in Greek, which means color, and soma in Greek, which means body. Initially, we don't know the diploid number of mammalian species because the chromosomes is crowded at the metaphase stage. But there's a lot of improvement as well as innovations of the cytogenetic analysis, such as the additions of culture sign to arrest cell metaphase stage, and as well as the use of hypotonic solutions for better chromosome spread. In 1956, it was determined that the diploid number of the chromosomes of human would be 46, and therefore it can be correctly counted as normal and abnormal chromosome number.
Conventional karyotyping.
Conventional karyotyping refers to the light microscopic presentations of the chromosomes set of a given cell.
The uses of conventional karyotyping.
The uses of it is to characterize the chromosomal complement of an individual and to study the chromosomal aberrations. This is an example of the karyotyping. So, karyotypings can be divided into two types. The first one, which be non-bending, which is the chromosome structures can be seen, but the bending of the chromosomes can't be seen, and as well as the bending of the chromosomes.
Principles of bending techniques.
The principle of it will start with G bending. It uses Giemsa stain. It yields series of lightly and darkly stained bands. The dark regions represent LT-rich region and the light regions represent GC regions. This is an example of the G bending at the right. So C bending also uses Giemsa staining, binds to constitutive heterochromatins, and it stains centromere. As you can see here, the centromere is darker than any parts of the chromosomes. Q bending, the pattern bends is very similar to G bending, but the staining used is quinacrine, that is the fluorescent dye.
So I'm gonna have a very good day. So today I'm going to explain to you guys about the procedure of the conventional cytogenetic analysis, which is a karyotyping. So the first thing is we're going to take a patient samples. So basically patient samples are from bone marrow and from peripheral blood. So first we're going to take a bone marrow aspiration. Sorry, this is okay. First we're going to take blood samples from bone marrow aspirations as well as peripheral blood. Which one is suitable? And after that, we're going to take the samples and put it into heparin tubes to prevent blood clotting. And then after that, we're going to put the samples into two flask tubes. Okay? So these two flask tubes, the first one is for the reactive methods, and the second one is for the synchronized methods. So these both flasks will have the same blood samples, antibiotics to prevent contaminations, PHA or phytoagglutinin, and as well as cosamide. Okay, phytoagglutinin is used to induce mitosis, and cosamide is to fix so that the cell will be in the metaphase phase. And for the synchronized method, first we're going to put a thymidine. This is where the difference between synchronized and reactive method. So thymidine will be used to elongate the chromosomes. Then after 16 hours of incubation with it, we're going to put a deoxycytidine, which is used to stop the elongation process. Then after that, we're going for the cell harvesting. Okay, harvesting will start after all of the incubations with the cosamide and everything. We're going to put it into a tube first. Then we're going to centrifuge it. Then we're going to add a KCl, okay, to have what we call the environment of hypotonic solutions. And then after that, we're going to incubate it for 30 minutes. So during the incubation of 30 minutes, we're going to prepare a fixed active. So fixed active, which is methanol to acetic acid in ratio of 3:1. So then after that, after we have 30 minutes, so we're going to put this methanol into the samples for prefix. Then we're going to centrifuge it for about 3 times. So centrifuge, remove the supernatant, adding again the prefix, then remove the supernatant again after centrifugations. Then we repeat it about 3 times. Then after that, you can proceed to sample dropping. So for the sample dropping, we're going to start with the samples. We put into a pipette. Then from a high place, we're going to drop the samples so that the cells would be burst, and showing its chromosomes. So we proceed with the staining. So in the staining, we're going to use a Leishman stains to see the chromosomes. Okay, and after that, we're going to view it under microscopes, and we're going to see an example of one spread of chromosomes from one cell. You can see here. Okay, so after that, we're going to do analysis about the patients, whether they have any what we call disorders and some sort of that. So that's all for me. Thank you. Hello, so today we're going to discuss about diseases that can be identified by karyotyping. So the first thing I'm going to discuss is about the autosomal chromosomal disorders. So I'm going to show you guys. Okay, first, the most common autosomal chromosomal disorders would be Down syndrome. So the patient with Down syndrome will have a trisomy 21, which means to have extra copy numbers of 21. And externally, the patients will have thick neck, short stature, and also almond eyes. And internally, the patient will have mental retardations, which mean they have IQ of around 20 to 70s, and heart defects, and as well as decreased fertilities. And so you can see here, this is the figure, which is almond eyes, thick necks, and short stature. So the next one is we're going to discuss about the Edwards syndrome. So Edwards syndrome, the patient have a trisomy 18. And externally, the patients will have a flexions of fingers, which is here, and a rocker bottom feet, which is a flexed big toe and prominent heels, and lot sets of ears. So the patient may have dysplastics or malformed ears. So internally, the patients will have a severe mental retardations and congenital heart disease, and that's why usually the patients will not last long. Okay, so the last one of the autosomal chromosomal disorders I'm going to talk is about the Patau syndromes. So patients with Patau syndromes have a trisomy 13, which means the same thing, extra chromosome number of 13. So externally, they will have a small eyes and head, and have usually a cleft palate in here, as you can see, and sometimes they have extra fingers, okay, and as well as a rocker bottom feet, the same as the Edwards syndromes. Okay. Okay. So then internally, the patient will have severe mental retardation, congenital heart defects, and eye, lung, and brain defects. And yeah, the same things. The sad thing is the patient won't live long. So the next one, going on to the sex chromosome disorders. So the first is Turner syndrome. So the patient with Turner syndromes have a short stature, ovarian dysgenesis, webbed neck, as you can see here, webbed necks, and cystic hygroma occurs around the ovarian, and the patients, which means will be infertile. And Turner syndromes will be the patients have only one X chromosomes. So yeah. Next one is Klinefelter syndrome, which is the patient will have 2 X and 1 Y. So the patients will have to externally have a tall stature, long arms and legs, okay, testicular atrophy, which is a small testicular size, it's gynecomastia, which has a breast development in here, and a female hair distributions. So and the patient is male, but have a tendency to grow a few chest hairs, more like a girl and less like a man, you know what I mean? And the last one I'm going to discuss is about triple X syndromes, which is the patients who have a super 3X. So, but triple X syndromes, you don't know who that person is unless you do a karyotyping, and the patients will usually is very healthy and very fertile. As you can see, so cute. So that's all from me.
Next, we are going to the advantages and the disadvantages of the karyotyping. We'll start with the advantages. It enables the view of a whole chromosomes, suitable when a specific anomaly is suspected, and act as a general diagnostic tool to detect additional chromosomes abnormalities such as cytomegaloblastic leukemia. For the disadvantages, it detects major structural abnormalities, labor-intensive, and highly dependent upon operator experience and skills. Thank you. Bye.